Hi, in this essay, we address an unanswered question about how HIV damages our immune system. Is CD38 involved in the loss of CD4 T cells in HIV infection? It is well known that immune deficiency follows the decline of CD4 T cells, which are the target of HIV. However, the picture is more complicated. Immune cells of people with HIV are chronically activated, and this activation also predicts the progression of HIV disease. This predictive capacity is very notable in T cells. Among the different surface proteins that we use to measure T cell activation, CD38 is very remarkable because it is independent of other activation markers, it is independent of viral load, and it is independent sometimes of CD4 T cell counts. So how could CD38 promote CD4 T cell death? Well, CD38 is an enzyme. It degrades NAD, and it produces CADPR and ADPR. In the setting of HIV infection, CD4 T cells overexpress CD38 with a decrease in their content of NAD. They additionally are exposed to a milieu with a lot of activity of CD38 and a low NAD concentration. This promotes the exit of NAD outside the cytoplasm. Now, NAD is essential for the function and for the integrity of mitochondria. So we can expect that a shortage of NAD compromises mitochondria and decreases the viability of CD4 T cells. This scenario is aggravated by CADPR and ADPR. They can be transported inside the cytoplasm, where they can activate calcium channels and increase calcium levels. High cytoplasmic calcium further stresses mitochondria, also promoting CD4 T cell death. All these processes have been demonstrated separately in different tissues and cell lines. However, the whole machinery required for their occurrence is present in CD4 T cells, particularly in HIV infection. Our model can be tested experimentally. For instance, we can determine if inhibiting CD38 reverses the lowered viability and proliferative capacity of CD4 T cells. We can also measure NAD contents of CD4 T cells from patients and controls, among other possible experiments. This is our proposal.